Hello everyone, my name is Dennis Motonga and I'm going to take you through the history of DNA. Welcome. So, first things first, we need to understand what DNA means. So, DNA is the definition of the word deoxyribonucleic acids, hence DNA. So, is the hereditary material in human and almost all living things. So, DNA is the reason why we resemble our parents, we resemble our siblings, and all that. So, it is in a shape that's like a twisted ladder or a spider staircase, and that's called double helix shape. So, uh, I divided my presentation into two parts. Number one, um, the early foundations and the carrier. So we're going to start with the first part where we'll be looking where we got this DNA from, because we're looking at the history of the DNA, and then um, basically see or identify the chemical basis of inheritance at the age of microscopy. And microscopy, for understanding, is the use of microscope to observe things that we can't see by our own eyes. So we're going to look at this scientist called Frederick Measure in 1869. So this photo is for that scientist, and this is the one who started the research for to look at the past from the surgical bandages from patients, and then he came up with the name nuclein. And in this, the substance was found was protein digesting resistant, like it wasn't being digested by the enzymes. And then the scientific community believed that this protein was so um, significant for the heredity, and heredity is the passing on of traits or characteristics from one person. So, defining the chemical structure, we had I had two sources that I will be looking at, and then the first one is Albrecht Kossel in 1881, who came up and who coined the nucleic acid as the deoxyribonucleic acid, and isolated the five nitrogenous bases from it. That is adenine, uracil, thymine, guanine, and cytosine. Then the second one is this one here called Phoebus Levin. He discovered three basic components of nucleotide. That is a sugar, a phosphate, and a base. So the second part is the search for the carrier. And as I said earlier, DNA can be carried from one person or one individual to another. So we have this Friedrich who did a research on the transformation of some molecules. So came up and found out that some molecules are carried information that survived heat killing, because he was testing on the heat killing mechanisms on different um, like for these for these for this photo right here that is on the left. You can see the rats there being injected with something, so others survived, others died. So this is the suggestion that was given out that the information there was an information that will be carried, and that or made the mouse to survive the heat killing. So definitive proof of the that the DNA is hereditary. So I had three sources here. As you can look at them, you had one with that DNA was a specific molecule responsible for Griffith transformation. Griffith transformation is that experiment that we just looked at about the mouse. Then we had the others talking about the DNA entered the host cell to replicate, confirming its true genetic material. So, for example, if we are born, a father gives me, let's say, half, and and the mother gives me half of the chromosomes so that I can born to the I can have the traits from both the father and the mother. In the next slide here we have this photo right here that is called photo 51 for Rosalind Franklin, and this photo revealed that helical structure and X-rays and precise dimensions that will allow the final model for the DNA. So Rosalind, through her work shared without her knowledge, it remains the most famous image in the biological, specifically this uh, genetics or biological history. We have in 1953, two scientists, one called James Watson and Francis Crick, proposed the double helix and anti-parallel helix model in Cambridge. And then this structure immediately suggested how DNA could replicate itself by splitting and using its strand as a template. So we have some sources here. For example, 1977, the Sanger, we had the first reading for the DNA sequence. 1983, invent a way to, that is Carl Smalls invented a way to amplify small DNA samples. We have a genome project that was completed in 2003, and then lastly here we have a tool for precise efficient gene editing that was discovered in 2012. So I want to thank you for watching this video with me. My name is Denim Stranga, and as we have explored together the history of the molecule that defines the people we are today. So thank you for watching this video. As you're enjoying your World Cup, I wish you all the best.